Okay, we'll start the class now. So today is the 8th problem solving session for the course Overview and Integration of Cellular Metabolism. The course instructors are Professor Ritri Beer and Professor Arindam Ghosh from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. So as you know the format, we have been discussing the assignment related questions. So today we'll discuss the questions related to phenylalanine, tyrosine, and tryptophan metabolism and different uh, products that we get from these amino acids, as well as how these amino acids are broken down. Okay, so I have prepared certain questions related to your assignment. We'll discuss all the questions and feel free to ask any doubt you have at any point. If you have any other doubt which I haven't covered in these questions, feel free to ask me. I'll try to explain uh, at my best capabilities. So, shall I start the class? Starting with the first question. Which enzyme converts phenylalanine to tyrosine? Option A, tyrosine hydroxylase. Option B, phenylalanine hydroxylase. Option C, dopamine decarboxylase. And option D, tryptophan synthase. Which enzyme converts phenylalanine to tyrosine? Tyrosine hydroxylase, phenylalanine hydroxylase, dopamine decarboxylase, or tryptophan synthase. You have answered option B, that is phenylalanine hydroxylase. That is the correct answer. So phenylalanine hydroxylase is the enzyme that converts phenylalanine into tyrosine. So this is the reaction, and this is actually the first reaction in the phenyl metabolism step. So first phenylalanine is converted into tyrosine, and then tyrosine is further metabolized in the body. So the first enzyme in this pathway is phenylalanine hydroxylase. As you can see here, phenylalanine uh, is hydroxylated at the para position by this enzyme to make tyrosine. So the difference between phenylalanine and tyrosine is this OH group at the para position. Para position, that means it is exactly opposite to this carbon, right? So this is the para position to with respect to this group. Okay, this would be the ortho position. Okay, so phenylalanine hydroxylase, it hydroxylates the phenylalanine at the para position to make tyrosine. So phenylalanine phenylalanine is hydroxylated at the para position, as I've already mentioned, by one atom of oxygen to produce tyrosine, and the other atom is reduced to water. So basically, if you see here, the oxygen molecule is converted to water. So you know, molecule of oxygen has two oxygen atoms, right? O2. So one oxygen is coming at the para position. This hydrogen that was there at the third carbon shifts to the fourth that was there at the fourth carbon shifts to the third carbon. So the hydrogen atom is transferred directly from C4 to C3 in the reaction. And this feature, since it was discovered by NIH, it is called NIH shift. And this reaction is irreversible, and it requires a very specific coenzyme, biopterin, which is structurally related to folate. So folate is a vitamin. So this biopterin is structurally related to folate, and phenylalanine hydroxylase basically is hydroxylating this particular carbon. So, but the hydroxylation is occurring at this position, but the hydrogen that was originally present at this carbon is shifting to the adjacent carbon. Okay, so that's how the reaction is proceeding, and one of the atom in the oxygen molecule is reducing into water molecule. Okay, I hope that's clear. Moving on to the next question now. Which cofactor is required for phenylalanine hydroxylase? Option A, NADPH. Option B, tetrahydrobiopterin. Option C, FAD. And option D, pyridoxal phosphate. Which cofactor is required for phenylalanine hydroxylase? NADPH, tetrahydrobiopterin, FAD, or pyridoxal phosphate? You have answered option B again, that is tetrahydrobiopterin. Yes, that is the correct answer. So, phenyl, as we saw in the previous slide also, phenylalanine hydroxylase requires a coenzyme or cofactor tetrahydrobiopterin for the reaction to proceed. So, the active form of biopterin is tetrahydrobiopterin. This type tetrahydrobiopterin is actually oxidized to dihydrobiopterin, that is the quininoid form. And tetrahydrobiopterin is then regenerated by an NADPH dependent dihydrobiopterin reductase. So one NADPH is oxidized into NADP plus, and in return tetrahydrobiopterin is reduced to dihydrobiopterin is reduced to tetrahydrobiopterin. Okay, and phenylalanine hydroxylases, this enzyme that we studied in the previous slide was it is present in liver. Okay, so this is how the regeneration of tetrahydrobiopterin occurs. So tetrahydrobiopterin is oxidized into dihydrobiopterin form, and when this oxidation reaction is occurring, simultaneously phenylalanine hydroxylase is hydroxylating the phenylalanine into tyrosine. Okay, and this conversion of tetrahydrobiopterin into dihydrobiopterin goes through an intermediate, that is pterin 4A carbinolamine. So this carbinolamine form is the intermediate form, and once this dihydrobiopterin is forming, it is again regenerated back to BH4, that is tetrahydrobiopterin, by dihydrobiopterin reductase, and it is a NADPH dependent enzyme. So dihydrobiopterin reductase, in this enzyme, the NADPH will be oxidized to NADP plus. In return, dihydrobiopterin will be reduced to tetrahydrobiopterin. Okay. So you see, along with phenylalanine hydroxylase, here two more enzymes are involved. One is pterin 4A carbinolamine dehydratase. So here one H2O is removed. So that's a dehydration reaction is occurring. So that is why dehydratase enzyme is there. And one reduction reaction is happening. So that's why a reductase is there. Okay. So here hydroxylation reaction is happening. So we have the enzyme hydroxylase. Then here dehydration reaction is happening. So we have the enzyme dehydratase. And here reduction reaction is happening. So we have the enzyme reductase. Okay, so depending on the reaction, the enzymes are named. I hope that's clear. Moving on to the next question now. Deficiency of phenylalanine hydroxylase causes which disorder? Deficiency of phenylalanine hydroxylase causes which disorder? Option A, alkaptonuria. Option B, albinism, option C, phenylketonuria, and option D, tyrosinemia. Deficiency of phenylalanine hydroxylase causes which disorder? Alkaptonuria, albinism, phenylketonuria, or tyrosinemia? So you have answered option C. That is phenylketonuria. That is the correct answer. So you saw phenylalanine hydroxylase, where it is involved. It is involved in the first reaction of the phenylalanine metabolism, that is the conversion of phenylalanine into tyrosine. And if this enzyme is deficient, the condition that we have is phenylketonuria. What will happen if an enzyme is deficient? That means substrate accumulation will start. So basically, phenylalanine is converted into tyrosine by this enzyme phenylalanine hydroxylase, right? So if this is not there, that means phenylalanine will start accumulating. The concentration of phenylalanine will increase and tyrosine will decrease, right? Because there's no enzyme that can convert phenylalanine into tyrosine. Such condition is called phenylketonuria, and the result that we get is hyperphenylalaninemia. Okay, that means the accumulation of phenylalanine in the body, in the blood. Okay, so it's a most common disease of amino acid metabolism, and due to deficiency of the phenylalanine, it occurs due to the deficiency of this enzyme phenylalanine hydroxylase. And this is a genetic disorder. And why does it happen? It happens due to a mutation. So a single mutation uh, occurs in the recessive allele of this gene, the phenylalanine hydroxylase gene. And the location of this mutation is on the long arm of the chromosome 12, locus 22. Okay, so you should remember this. This could come as an MCQ question as well. So you should remember this. And it's an autosomal recessive disorder. So if both the parents are carrier, then 25% chances are there that the progeny will have this disease. If one of the parent is carrier, then the chances become less, but then the progeny could be carrier as well. So it's important to do the uh, genetic analysis before having kids, so that you know that if there's a genetic defect, if there's an autosomal recessive genetic defect present in both the partners' gene pool, then there's a chance that the progeny might get these diseases. Okay. And I told you that it results in accumulation of phenylalanine, leading to hyperphenylalaninemia.
Ma'am, one doubt, ma'am.
Yeah.
There's always a doubt that that Mosi order in this phenylketonuria is due to the metabolite, whether it is due to phenyl lactate or phenyl acetate, or
Yes. Yes. Yes.
there's always a debate is there. But the correct answer for this Mosi order is due to uh, which of these metabolites, ma'am?
It is due to the phenyl pyruvate. I'll come to that. I have a question for that also. So basically,
Oh,
this phenylalanine, when it keeps getting accumulated, instead of, because phenylalanine now cannot be converted into it cannot be converted into tyrosine, right? So there are other enzymes. There are some aminotransferases that converts this phenylalanine into phenyl pyruvate. Okay, and those compounds, they are secreted in urine, sweat, and that gives that odor. Phenylalanine does not give any odor, but the metabolites that are converted from phenylalanine, because now the phenylalanine cannot proceed to become tyrosine, right? This is not happening. So what happened? There are other enzymes in the body. So they will, since the concentration of phenylalanine is increasing, those other enzymes will start taking up the phenylalanine and convert it into phenylalanine pyruvate. Okay, and then there are other metabolites that gets converted from phenylalanine pyruvate, and those metabolites are actually responsible for the mousy odor. Clear?
Okay, ma'am.
So moving on to the next question. What is the presently what is the primary treatment for phenylketonuria? Option A, administration of phenylalanine rich diet. Option B, supplementation with tyrosine. Option C, restriction of phenylalanine in the diet. And option D, gene therapy. What is the present treatment for phenylketonuria? Option A, administration of phenylalanine rich diet. Option B, supplementation with tyrosine. Option C, restriction of phenylalanine in the diet. And option D, gene therapy. You are saying option C, restriction of phenylalanine in the diet. So that is the correct answer. So basically, phenylalanine is getting in case of phenylketonuria, phenylalanine is getting accumulated, right? So if you restrict the phenylalanine content in the diet, then automatically the total amount of phenylalanine in the body will be low. So even if you don't have the enzyme that converts phenylalanine into tyrosine, uh, it won't lead to major problems that are
But
associated with phenyl.
ma'am, but the
Yes.
only function of it is to get converted to tyrosine, no, ma'am. So what the source for tyrosine? It is an essential amino acid, no, ma'am. Then becomes.
Yes, so you have to take tyrosine. So anyway, like even for phenylketonuria, you cannot completely remove phenylalanine from diet. So you have to take tyrosine and phenylalanine in diet. These are essential amino acids. They should be there in the diet. But for cases where phenylalanine cannot be converted into tyrosine, the amount of phenylalanine in the diet should be restricted. You can take
So that
supplement.
comes the primary treatment, first option. Restriction of phenylalanine is the first option. Then
Yes,
second will be supplementation with tyrosine.
supplementation of tyrosine would be the secondary treatment. Like primary treatment would be to restrict the phenylalanine first. Then,
Okay.
then supplementation with tyrosine is not actually a treatment for phenylketonuria, because the problems that are associated with phenylketonuria are because of the accumulation of phenylalanine. It is not because of the depletion of tyrosine. Like major symptoms that we see in phenylketonuria, they are due to the accumulation of phenylalanine. So first we have to solve that problem. First we have to make sure that amount of phenylalanine in the body is in limit. And then like as a result, now the body needs tyrosine, so you can supplement tyrosine in the diet and you can take tyrosine supplements. So that is a different issue, but then it's not the symptoms of phenylketonuria are not because of the lack of tyrosine. It is actually because of the major symptoms are due to phenylalanine accumulation also, right? So the primary treatment has to be restriction of phenylalanine. After that, supplementation with tyrosine would be.
Okay, ma'am. Thank you.
Moving on to the next question now. What is the main clinical manifestation of untreated phenylketonuria? What is the main clinical manifestation of untreated phenylketonuria? First is mental retardation. Second is osteoporosis. Third option, hemolytic anemia. And fourth option is uh, hyperuricemia. What is the main clinical manifestation of untreated phenylketonuria? Option A, mental retardation. Option B, osteoporosis. Option C, hemolytic anemia. And option D, hyperuricemia. You are saying option A, that is mental retardation. That is the correct answer. So, why does mental retardation occur in phenylketonuria? Do you have any idea? Can you tell me why mental retardation is a clinical manifestation of phenylketonuria? Yeah, so tyrosine depletion, what is tyrosine depletion? What does tyrosine depletion do? Why tyrosine is important for uh, our central nervous system, or why tyrosine depletion is causing mental retardation? Yes, the hormones that are derived from it are low. So those hormones are actually critical for the central nervous system's functioning. So tyrosine is responsible for certain neurotransmitter synthesis of certain neurotransmitters. So that those are hampered because of low tyrosine. Yeah, catecholamines are there. Along with that, like coming to your first question, that was why phenylalanine restriction is important. Phenylalanine itself is so basically phenylalanine is a neutral amino acid, right? And it blocks the neutral amino acid transporters that are there in the uh, brain. So we have different new uh, neutral amino acid transporters in the brain, and those are required for neurotransmitter synthesis. Okay. And if phenylalanine is in large quantity, it will kind of block all the transporters, and it won't allow the other neutral amino acids to uh, cross these transporters and reach the brain for the neurotransmitter synthesis. And that can lead to uh, basically that can lead hinder the development in the brain, causing intellectual disability. And phenylalanine, as I've already mentioned, is involved in tyrosine synthesis, and tyrosine uh, lack of tyrosine can lead to hampered synthesis in the catecholamines. Dopamine is a very important neurotransmitter that is produced by tyrosine, and if tyrosine is depleted, dopamine will be depleted, and that can also lead to mental retardation states. Okay, so these are the main reasons. So, like when you were asking in the previous question why phenylalanine is important, this is why phenylalanine has to be restricted. Otherwise, untreated, in case of untreated phenylketonuria, mental retardation and other CNS symptoms are very common if this phenylketonuria is left untreated. Okay. So I hope this part is clear, right? So phenylalanine being a large neutral amino acid, they have certain, brain has certain large neutral amino acid transporter through which these amino acids cross the blood-brain barrier. So if all the transporters are occupied by phenylalanine, the other amino acids won't be able to cross the blood-brain barrier. And those amino acids are also required in the brain for these protein and neurotransmitter synthesis. And if that is not happening properly, there could be different developmental issue in the brain leading to mental retardation. Okay. Moving to the next question now. Both phenylalanine and tyrosine are option A, glucogenic, option B, ketogenic, option C, glucogenic and ketogenic, and option D, neither. Both phenylalanine and tyrosine are option A, glucogenic, option B, ketogenic, option C, glucogenic and ketogenic, and option D, neither. Both phenylalanine and tyrosine are glucogenic, glucogenic and ketogenic, or neither. You're saying option C, glucogenic and ketogenic. Can you tell me why?
Because the end product of the whole metabolism is fumarate and acetoacetate, where fumarate can enter into the TCA cycle, and acetoacetate is a ketone, is a ketogenic product.
Yes. So
Both.
if you remember from the previous session, acetoacetate is the ketone body. So when we studied the ketone body synthesis and metabolism, we saw that the acetoacetate was the primary ketone body, right? So it enters the ketogenic pathway, ketogenic metabolism, and fumarate is the glucogenic substrate. It enters the TCA cycle, which is the glucogenic uh, metabolic pathway. Okay, so phenylalanine and tyrosine both are glucogenic and ketogenic. So phenylalanine and tyrosine metabolism is basically a common pathway. Phenylalanine first gets converted into tyrosine, and then tyrosine gets converted into fumarate and acetoacetate via multiple steps. And that fumarate and acetoacetate both, so fumarate is the glucogenic substrate and the acetoacetate is the ketogenic substrate. So option C is the correct answer. Basically, the final products, one is glucogenic, one is ketogenic. Okay, moving to the next question. Elevated phenylpyruvic acid in urine is the characteristic of. So you have already asked this question. So now we'll discuss why we were getting the mousy smell. So basically, elevated phenylpyruvic acid in urine is characteristic of option A, heart nerve disease, option B, albinism, option C, phenylketonuria, and option D, tyrosinemia. Option A, heart nerve disease, option B, albinism, option C, phenylketonuria, and option D, tyrosinemia. We are saying option C. We have already discussed this. So in phenylketonuria, the phenylalanine that is getting accumulated, it actually gets converted into phenylpyruvic acid. So this is the pathway. So phenylalanine transfer its amino group to pyruvate, and pyruvate becomes alanine, and phenylalanine becomes phenylpyruvate. This phenylpyruvate can further get converted into phenyl acetate and phenyl lactate. Okay. And these phenyl lactate, phenyl acetate, and phenyl pyruvate, they give the mousy or musty smell to the breath, skin, and urine. Okay. So phenylalanine via aminotransferase. So if you remember from the previous class, we studied about aminotransferase enzymes, right? What were aminotransferases? What do aminotransferase do? They basically take the amino group from one amino acid and transfer it to a keto sugar, alpha keto sugar, and that keto acid will get converted into amino acid, and the amino acid will get converted into a keto acid. So if you see here, phenylalanine, it has an amino group, it's an amino acid, so it has an amino group, it has an acid group. So this phenylalanine, this will transfer the amino group to a keto acid, that is pyruvate in this case. So pyruvate is a keto acid, it has a keto group and an acid group, so it's a keto acid. This amino group will be transferred to pyruvate. Now, if instead of this ketone functional group, if we have a amino group attached to this carbon, it will become alanine, right? So pyruvate gets converted to alanine. And if I now add a keto group instead of this amino group, what will I get? I will get phenyl pyruvate. Okay, this is the pyruvate, and the pyruvate has a, so you can see the structure of pyruvate here, right? So pyruvate has a substitution of a phenyl group. A benzene ring is attached to this phenyl, so it becomes phenyl pyruvate. And this phenyl pyruvate can further either undergo decarboxylation, so the CO2 will get removed, and we'll have CH2CO minus, so phenyl acetate, or it can undergo or it can become phenyl lactate. So this ketone can get reduced to hydroxyl group. So instead of keto group, now we have a hydroxyl group. So it becomes phenyl lactate. This is the lactate. So all these compounds, phenyl lactate, phenyl acetate, and phenyl pyruvate, they all are secreted in the urine, and they give the mousy smell to the urine, and it also gives the mousy smell to the breath and skin. Is this clear? Do you now understand
Yes, ma'am.
what you were asking in the previous question about the
Yes.
mousy smell? Okay, then I'll move on to the next question. Which enzyme defect leads to Rickner-Hanhart syndrome? Which enzyme defect leads to Rickner-Hanhart syndrome? Option A, tyrosine transaminase. Option B, fumarate acetoacetate hydroxylase. Option C, phenylalanine hydroxylase. Option D, homogentisate dioxygenase. Which enzyme defect leads to Rickner-Hanhart syndrome? Tyrosine transaminase, fumarate acetoacetate hydroxylase, phenylalanine hydroxylase, and homogentisate dioxygenase. Which enzyme defect leads to Rickner-Hanhart syndrome? Option A, tyrosine transaminase. Option B, fumarate acetoacetate hydroxylase. Option C, phenylalanine hydroxylase. And option D, homogentisate dioxygenase. So you have answered option A, that is tyrosine transaminase. That is the correct answer. So tyrosine, after phenylalanine gets converted into tyrosine, the tyrosine is further metabolized to get to form fumarate and acetoacetate. We saw that in the previous question, right? So in that pathway, the first step is the aminotransferase step, where amine group is transferred to a keto acid, like I explained in the previous slide. So tyrosine transaminase or tyrosine aminotransferase, these two names can be used interchangeably. So this enzyme actually converts the tyrosine, deaminates the tyrosine. So It deaminates the tyrosine to make para-hydroxyphenyl pyruvate, and tyrosine gives the amino group to the alpha ketoglutarate to become glutamate. Okay, so alpha ketoglutarate converts into glutamate and tyrosine converts into para-hydroxyphenyl pyruvate. And this reaction is catalyzed by tyrosine aminotransferase or uh, tyrosine transaminase. This, if there is a deficiency or a defect in this enzyme, as a result, what will happen? Tyrosine will not be able to uh, further metabolize and tyrosine will start getting accumulated. And the condition that is developed due to the defect in this enzyme is the tyrosinemia type 2. Tyrosinemia, any defect in the tyrosine metabolism is classified under tyrosinemia, where tyrosine is getting accumulated because it is not broken down into the next compounds in the metabolic pathway, right? So tyrosinemia. So this is the tyrosinemia type 2, where the defect is in the first enzyme, tyrosine transaminase. This defect is also known as oculocutaneous tyrosinemia, or it is known as Rickner-Hanhart syndrome. It results in blockage in the routine degradative pathway of tyrosine, as you can see, and it is characterized by skin, hyperkeratosis of palm, and eye lesions. Painful corneal lesions are observed, as well as some neurological problems that are mental retardation and all are observed in this in case of tyrosinemia type 2. So basically, in any of the tyrosinemia defects, like irrespective of where the defect, at what step the defect is, the result would be tyrosine accumulation, and it can lead to different clinical manifestations. So tyrosinemia type 2, that is the Rickner-Hanhart syndrome or oculocutaneous tyrosinemia, it is characterized by skin hyperkeratosis of palm and eye lesions. So that's why it's called oculocutaneous. So it affects the skin and eyes. So uh, basically, you see thick skin developing on the palm and feet, and even there are corneal lesions present in the eyes, and in some cases mental retardation is also observed. Okay, so in the tyrosine metabolism, the defect in first enzyme leads to tyrosinemia type 2 or oculocutaneous tyrosinemia. Moving on to the next question now. Tyrosinemia type 1 is due to deficiency of homogentisate oxidase, fumarate acetoacetate, tyrosine aminotransferase, 4-hydroxyphenyl pyruvate dioxygenase. Tyrosinemia type 1 is due to the deficiency of option A, homogentisate oxidase, option B, fumarate acetoacetate, option C, tyrosine aminotransferase, and option D, 4-hydroxyphenyl pyruvate dioxygenase. You are saying option B, that is fumarate acetoacetate. That is the correct answer. So tyrosinemia type 1 is caused due to the deficiency of fumarate acetoacetate. So again, in the tyrosine metabolism, the last step of the metabolism, where the fumarate acetoacetate is broken down into fumarate and acetoacetate, that breakdown is done by a fumarate acetoacetate enzyme. It's a hydroxylation reaction. One H2O molecule is introduced, hydrolysis reaction, sorry, not hydroxylation. It's a hydrolysis reaction. And defect in the enzyme fumarate acetoacetate leads to tyrosinemia type 1. So we studied about tyrosinemia type 2. This is next defect, that is tyrosinemia type 1. It causes liver failure, rickets, renal tubular dysfunction, and polyneuropathy. And in case of tyrosinemia type 1, tyrosine and its metabolite, they are excreted in the urine. So naturally, if you don't have this enzyme, tyrosine cannot be broken down into fumarate and acetoacetate. The last step cannot happen. So tyrosine as well as the other intermediate metabolites like para-hydroxyphenyl pyruvate and then fumaryl acetoacetate, these will get accumulated. So tyrosine and its metabolite, they will be excreted in the urine because they are not further metabolized. Then it can lead to both acute and chronic manifestation. Under the acute manifestation, we have hepatic and renal failure leading to death within 1 year. So it can cause liver and kidney failure. And under chronic manifestations, you have diarrhea, vomiting, cabbage-like odor due to the production of these organic acids, and vitamin D resistant rickets. So this is a very prominent manifestation of tyrosinemia type 1. And because this fumaryl acetoacetate is not broken down into fumarate and acetoacetate, this fumaryl acetoacetate can actually get converted into succinyl acetoacetate, and succinyl acetoacetate can get converted into succinyl acetone. Now, due to the increasing level of fumaryl acetoacetate, eventually succinyl acetoacetate will increase, and this succinyl acetoacetate can be easily diagnosed. So if in a patient sample, succinyl acetone is high in the levels of succinyl acetone is high, then the patient is diagnosed with tyrosinemia type 1. So this is a very prominent diagnostic marker, succinyl acetone, because fumaryl acetoacetate, which is naturally converted to fumarate and acetoacetate by the enzyme fumaryl acetoacetate, since that is not present or that is defective, this fumaryl acetoacetate gets converted into succinyl acetoacetate and further to succinyl acetone. The succinyl acetone levels is a diagnostic feature for tyrosinemia type 1. So this is important for you to remember, like which compound is actually measured to diagnose tyrosinemia type 1. It is succinyl acetone. And succinyl acetone has the property of inhibiting the delta aminolevulinic acid metabolism, which is involved in heme synthesis. So when we read about heme synthesis, heme metabolism, at that time we will see this delta aminolevulinic acid, and delta aminolevulinic acid metabolism is inhibited by succinyl acetone. That means in case of tyrosinemia type 1, where fumaryl acetoacetate is not working, succinyl acetone will keep getting accumulated, as well as delta aminolevulinic acid will keep getting accumulated. Okay. And there will be a heme depletion as well. Okay. So these are different manifestation and different features of tyrosinemia type 1. Is that clear?
Yes, ma'am.
Moving to the next question now. The main defective enzyme in alkaptonuria is option A, homogentisate synthase, option B, homogentisate phosphorylase, option C, homogentisate oxidase, Option D, homogentisate lyase. And option E, homogentisate carboxylase. The main defective enzyme in alkaptonuria is homogentisate synthase, homogentisate phosphorylase, homogentisate oxidase, homogentisate lyase, and homogentisate carboxylase. You have answered option C, that is homogentisate oxidase. That is the correct answer. So alkaptonuria, like, it's also a form of tyrosinemia. So like in tyrosinemia type 2, we had aminotransferase deficiency or defect. Then in type 1, we had fumarate acetoacetate defect. And in alkaptonuria, we have the defect in the enzyme homogentisate oxidase. So this is the whole pathway actually of the phenylalanine metabolism. So phenylalanine is converted into tyrosine by the enzyme phenylalanine hydroxylase. Defect in this enzyme is leading to phenylketonuria. The next step is the amino group transfer of tyrosine. This leads to parahydroxyphenylpyruvate formation. So defect in the enzyme tyrosine aminotransferase leads to tyrosinemia type 2. Then the next step is the conversion of hydroxyphenylpyruvate into homogentisate. This is done by the enzyme parahydroxyphenylpyruvate dioxygenase. It's a dioxygenase. It adds the 2 oxygen molecule. It basically adds 2 OH group. And the defect in the enzyme parahydroxyphenylpyruvate dioxygenase is known as tyrosinemia type 3. Then homogentisate is converted into melyl acetoacetate by the enzyme homogentisate 1,2-dioxygenase, and the defect in this enzyme leads to alkaptonuria. Then we have melyl acetoacetate isomerization into fumarate acetoacetate, and then fumarate acetoacetate is hydrolyzed into fumarate and acetoacetate, and we have already seen the defect in this enzyme is leading to tyrosinemia type 1. Okay, so right now we are talking about alkaptonuria. So alkaptonuria, it is caused due to the defect in the homogentisate 1,2-dioxygenase enzyme that converts homogentisate into melyl acetoacetate. As a result, since this enzyme is defective, homogentisate will keep getting accumulating in the tissues and bloods, and it is excreted in urine. On standing, the homogentisate gets oxidized to corresponding quinones, which polymerizes to give black or brown color. So if we keep homogentisate for some time undisturbed, it will turn into a black or brown color due to the quinone formation. So these OH will get converted into double bond O ketone groups. So since homogentisate is excreted in the urine, if you keep the urine for some time, it can turn into this black color. So you can see the difference. And this is a clear indication of homogentisate presence in urine, and this eventually leads to the conclusion or it's a diagnostic feature for alkaptonuria. Okay, so urine resembles coke in color. So you know how coke looks. So that color, the urine turns into that color. Homogentisate gets oxidized by polyphenol oxidase to benzoquinone acetate. So this is homogentisate. It is oxidized by polyphenol oxidase to form benzoquinone acetate, which undergoes polymerization to produce a pigment called alkaptone body. So inside the body, so this is happening outside, okay? In the urine, homogentisate is excreted out, And if you keep the urine for some, if you collect the urine and keep it for some time, you will see that it has turned black because of the oxidation, the air oxidation, auto-oxidation of homogentisate into quinones. But inside the body also, homogentisate is oxidized by polyphenol oxidase to form benzoquinone acetate, and these benzoquinone acetate can actually undergo polymerization to form a pigment called alkaptone bodies, and these alkaptone bodies can deposit in the connective tissues, bones, various organs, resulting in condition called ochronosis. So this is the vertebral column, and you can see the black pigment accumulated between the vertebral column. These are all these are all indication of ochronosis or deposition of the polymerized benzoquinone acetate, which are called the alkaptone bodies. So this is the deposition of the alkaptone bodies, and this is condition is called ochronosis. Arthritis can also happen due to the deposition of this alkaptone body in the joints. And treatment, basic treatment is by consumption of protein diet with relatively low phenylalanine content. So if you have a diet of low phenylalanine, there will be lesser tyrosine and there will be lesser amount of homogentisate forming, and these clinical manifestations will be reduced. Okay. So again, phenylalanine is an essential amino acid. So small amount is required. That's why it's mentioned relatively low phenylalanine content. It's not written that you have to remove phenylalanine. You have to restrict the diet. You don't have to remove the phenylalanine or tyrosine in that matter. We never we can never eliminate the phenylalanine and tyrosine from our diet because these are essential amino acids. These are not synthesized by our body. We have to take these amino acids in our diet because that's the only source of these amino acids. But if any of the enzymes are defective in the metabolism of these amino acids, we take them in limited quantities so that how much ever is required, it is going in the other pathways, and for the metabolism, it's not getting accumulated at any step. I hope that's clear.
Yes, ma'am. It's clear, ma'am.
Moving to the next question. Biologically important compounds synthesized from tyrosine are all except. Well, what are the compounds that are synthesized by tyrosine, and which of the following is not synthesized by tyrosine? Biologically important compound that are synthesized from tyrosine are all except. So you have to tell which is the compound that is not synthesized by tyrosine. Option A, epinephrine. Option B, norepinephrine. Option C, dopamine. Option D, melatonin. And option E, triiodothyronine. Biologically important compounds synthesized from tyrosine are all except option A, epinephrine, option B, norepinephrine, option C, dopamine, option D, melatonin, option E, triiodothyronine. You have answered option D, that is melatonin. That is the correct answer. So melatonin basically is synthesized by serotonin, that is synthesized from tryptophan. We will see that in subsequent questions. Melanin is actually synthesized by tyrosine. That's a pigment that is present in the skin that is responsible for the skin coloration. So melatonin and melanin, these are two different compounds. Melanin is the pigment that is synthesized by tyrosine. Melatonin is synthesized by tryptophan. Okay, so you have to be very careful. Other than that, epinephrine, norepinephrine, dopamine, these are all catecholamines. These are synthesized by tyrosine. triiodothyronine, it's a intermediate in the process of thyroid metabolism, and it is also synthesized by tyrosine. So it's basically a iodized form of tyrosine. Okay, so triiodothyronine. I hope that's clear. I'll move on to the next question. Conversion of tyrosine to dopa needs all except what is not required for the conversion of tyrosine to dopa. Dopa is dihydroxyphenylalanine. Okay. So tyrosine is basically para-hydroxyphenylalanine, right? So if you add one more OH to tyrosine, it will become dopa. So conversion of tyrosine to dopa needs all except option A, NADPH, option B, molecular oxygen, option C, tyrosine hydroxylase, option D, tetrahydrobiopterin, and option E, PLP. Conversion of tyrosine to dopa needs all except option A, NADPH, option B, molecular oxygen, option C, tyrosine hydroxylase, sorry, option D, tetrahydrobiopterin, and option E, PLP. So you have answered option E, that is PLP. That is the correct answer. So tyrosine conversion of tyrosine to dopa is done by the enzyme tyrosine hydroxylase, and it has the cofactor tetrahydrobiopterin. It's attached, similar to the reaction of phenylalanine hydroxylase, where phenylalanine was converted to tyrosine. There also same reaction was happening, right? So that also required a molecular oxygen, NADPH, and tetrahydrobiopterin. Similarly, in this reaction also. So this reaction is very similar to that reaction. If you see, in case of phenylalanine hydroxylase, phenylalanine was converted to tyrosine, right? And so basically phenylalanine had the benzene ring, and this hydroxyl group was attached to the phenylalanine to make it tyrosine. Now, in this reaction, if you see, one more hydroxyl group is attached at this position. So in this position, there is only one hydrogen, and then one hydroxyl group is attached at this position to make it dihydroxyphenylalanine. So now it's a phenylalanine with two hydroxyl groups. So it's dihydroxyphenylalanine. So this reaction is very similar to the reaction of phenylalanine to tyrosine. The enzyme is tyrosine hydroxylase. Like in this case, we had phenylalanine hydroxylase. In this case, we have tyrosine hydroxylase. All the cofactors that are required are same. We need a molecular oxygen, we need NADPH, and we need tetrahydrobiopterin. And this reaction occurs in mitochondria. So this is also important. This the location of this reaction, it occurs in mitochondria. Okay, so it's a part of catecholamine synthesis pathway. So from tyrosine, different catecholamines are synthesized like dopamine, epinephrine, norepinephrine. So this reaction, the conversion of tyrosine into dopa, this is a part of catecholamine synthesis. Tyrosine is hydroxylated to 3,4-dihydroxyphenylalanine, It is a rate limiting step and requires tetrahydrobiopterin as coenzyme. So this is the whole pathway of catecholamine synthesis. So tyrosine is converted into DOPA by tyrosine hydroxylase. By tyrosine hydroxylase, tyrosine is converted into DOPA where one OH group is added. After that, DOPA decarboxylase, the enzyme DOPA decarboxylase basically decarboxylates this dopamine to form dopamine. So this carbon dioxide is removed. Decarboxylation reaction occurs. The cofactor for this enzyme is PLP. Dopa decarboxylase. The cofactor is PLP, and the carbon dioxide is removed. So we are just left with CH2, CH2, NH2, and this molecule is called dopamine. Okay, so we have the dihydroxyphenylalanine, DOPA, and from DOPA we get dopamine. This dopamine is further converted into norepinephrine and then epinephrine. So norepinephrine from DOPA is catalyzed by the enzyme dopamine hydroxylase, and this enzyme is a copper dependent enzyme. It requires copper ions and vitamin C. Okay, and at this position one OH is added. Then norepinephrine is methylated to form epinephrine. So the amine group will be methylated, and the methyl group is obviously transferred from S-adenosine methionine, and the enzyme is N-methyl transferase. So it is transferring a methyl group from SAM to form SAH, and we get epinephrine from norepinephrine. Okay, so tyrosine is converted into DOPA by tyrosine hydroxylase. Then DOPA is converted into dopamine by DOPA decarboxylase. Then dopamine goes to norepinephrine by dopamine hydroxylase. And then norepinephrine goes to epinephrine by N-methyl transferase. The synthesis of catecholamine is clear. If you have any doubt in this pathway, let me know. Is this clear?
Yes, ma'am, it's clear, ma'am.
Oh. Okay. Then we can go to the next question, that is, Parkinson's disease is marked by a lack of which chemical in the brain? Parkinson's disease is marked by a lack of which chemical in the brain? Option A, serotonin. Option B, GABA. Option C, dopamine. And option D, norepinephrine. Parkinson's disease is marked by a lack of which chemical in the brain? Option A, serotonin. Option B, GABA. Option C, dopamine. And option D, norepinephrine. You've answered option C. That is dopamine. That is the correct answer. So lack of dopamine actually leads to Parkinson's disease, and dopamine is important neurotransmitter. So lack of dopamine leads to a lot of clinical symptoms, and ultimately it causes the disease, Parkinson's disease. It's a very serious disease. and it's generally identified at a very old age. So Parkinson's disease is due to degeneration of the dopaminergic neurons. So the neurons, they require the dopamine, and then there's a lack of dopamine or decrease in dopamine production that leads to Parkinson's disease. Dopamine is involved in imitation of voluntary movements and the reward response reflexes. And it is also produced by hypothalamus, where it inhibits the synthesis of prolactin. And what are the effects of this disease? Due to the lack of dopamine production, there's a slowness of movements, there's rigidity of the muscle, repeated involuntary movements are called tremors. So patient gets tremors, they kind of their hands start shaking, they have kind of repeated involuntary movements. Patient often have an expressionless face where the muscles become rigid, okay? And then there's decreased eye blinking, flexion of head, stooping posture. These are very common, like typical symptoms of Parkinson's. Then sleep disturbances, change in mood, behavior due to depression, dementia, all these comes on as a manifestation of Parkinson's. Then debilitating conditions are there. Okay. Is this clear? Basically, Parkinson's make the patient very weak due to the lack of dopamine. Next question is, why is dopamine itself not used to treat Parkinson's disease? Now we saw that Parkinson's is caused due to the lack of dopamine, but we don't administer dopamine directly to the patient. We don't give them dopamine directly. Why is it so? Why do we not use dopamine directly to treat Parkinson's disease? Option A causes severe side effects. Option B, it cannot cross the blood-brain barrier. Option C, it is rapidly metabolized. And option D, it blocks the dopamine receptors. Why dopamine is not used to treat Parkinson's disease? Option A, it causes severe side effects. Option B, it cannot cross the blood-brain barrier. Option C, it is rapidly metabolized. And option D, it blocks dopamine receptors. You are saying option B, it cannot cross the blood-brain barrier. That's the correct answer. That's why we don't administer dopamine. Instead, we administer the dopa as a drug. And so if you notice the location of each step, each reaction happening in this pathway, in this catecholamine synthesis pathway, you will understand why dopamine is not given. So basically, tyrosine conversion of tyrosine into dopa occurs in mitochondria. Tyrosine hydroxylase is present in mitochondria. It converts the tyrosine into dopa. Then the dopa comes in cytosol, and dopa decarboxylase is present in the cytosol, where dopa is converted into dopamine. Once dopa gets converted into dopamine in the cytosol, then the dopamine enters the chromaffin granules of the pheochromocytes or granulated vesicles of brain cells or nerve endings. Okay, dopamine enters these chromaffin granules, and there the dopamine hydroxylase converts dopamine into norepinephrine. This norepinephrine again comes out in the cytosol, where the methyl transfer occurs to form epinephrine, and epinephrine again enters the chromaffin granules and stored in the chromaffin granules. So you see the location is very important. So if we administer dopamine directly, it cannot enter the brain directly because it has to go through this pathway. So when we administer dopa can easily enter the cytosol, and then there it can get converted into dopamine, and that dopamine can enter the uh, nerves or enter the promafin granule, where it can get converted into epinephrine and norepinephrine. Do you understand? So that's why dopamine is not administered during Parkinson's, since although the disease is due to the lack of dopamine, instead of administering dopamine, we administer one step, we go one step above in the pathway, and we administer the substrate of dopamine, or we can say we administer the starting material of dopamine, that is dopa. So dopamine is forming from dopa. So if we give dopa, our body will convert dopa into dopamine, and then dopamine can enter the brain. Is this clear? Okay, moving to the next question now. The degradation products of epinephrine is or are, so multiple answers could be correct in this case. Degradation products of epinephrine, option A, vanillyl mandelic acid, option B, COMT, that is catechol O-methyl transferase, option C, metanephrine, option D, dopamine, option E, selegiline. Degradation product of epinephrine, vanillyl mandelic acid, option B, COMT, that is catechol methyl transferase, option C, metanephrine, option D, dopamine, option E, selegiline. You have written option A and C. That is vanillyl mandelic acid and metanephrine. That is the correct answer. So vanillyl mandelic acid and metanephrine are the degradation products of epinephrine. So this is the degradation pathway of epinephrine. So once norepinephrine is converted into epinephrine by a methyl transfer, epinephrine gets converted into metanephrine by another methyl transfer, that is catechol O-methyl transferase. So this is the catechol, so 2OH in the phenyl ring. This forms the catecholic group, and in one of the hydroxyl group of the catechol, the methyl group is transferred. So here you can see. So here you can see this is the OH, and instead of OH, now a methyl group is transferred. So this transfer is occurring in the catechol O group. So catechol O methyl transferase, COMT is the enzyme involved. Again, the methyl group is taken from SAM, S-adenosine methionine. So methyl group will be transferred, and the product that is forming is called metanephrine. So epinephrine gets converted into metanephrine by catechol O-methyl transferase. So this is the first product. Then metanephrine gets converted into vanillyl mandelic acid, VMA, by the enzyme monoamine oxidase. So it's an oxidation reaction, monoamine oxidase. What will it do? It will remove this part. Okay. So we have CHOH, CO minus, that is the acidic group. So this is oxidized into vanillyl mandelic acid. The half-life of epinephrine is 2 to 5 minutes. So half-life of epinephrine is 2 to 5 minutes, and it is very quickly degraded into these products, metanephrine and vanillyl mandelic acid. Epinephrine is catabolized in tissue by catechol O-methyl transferase to metanephrine. Then MAO, that is monoamine oxidase, will oxidatively deaminate the metanephrine. Major end product is 3-hydroxy-4-methoxy mandelic acid or vanillyl mandelic acid. Normally, VMA is excreted 2 to 6 mg per 24 hours. VMA is increased in pheochromocytoma and neuroblastoma. So these are the two diseases where VMA is the amount of VMA is very high. Pheochromocytoma and neuroblastoma. In both cases, the VMA secreted or VMA excreted is much higher than 2 to 6 mg. Okay, so normally in 24 hours, 2 to 6 mg of VMA is excreted out. Epinephrine is degraded into metanephrine, and then metanephrine is degraded into vanillyl mandelic acid or 3-hydroxy-4-methoxy mandelic acid. I hope that's clear.
Yes, ma'am. It's clear, ma'am.
Okay. Moving on to the next question now. Which amino acid is the precursor for thyroid hormone synthesis? Which amino acid is the precursor for thyroid hormone synthesis? Option A, tryptophan. Option B, tyrosine. Option C, alanine. And option D, proline. Which amino acid is the precursor for thyroid hormone synthesis? Tryptophan, tyrosine, alanine, proline. Which amino acid is the precursor for thyroid hormone synthesis? Option A, tryptophan. Option B, tyrosine. Option C, alanine. And option D, proline. You're saying option B, that is tyrosine. That is the correct answer. So this is the thyroid synthesis. So tyrosine is basically iodinated to form the thyroid hormone. So iodination of tyrosine ring occurs to produce mono and diiodotyrosine, from which triiodothyronine and thyroxine, that is tetraiodothyronine, are synthesized. The protein thyroglobulin undergoes proteolytic breakdown to release the free T3 and T4. So there's a protein thyroglobulin. The tyrosine residues in this protein thyroglobulin are iodinated. The phenyl ring of the tyrosine is actually iodinated. So one iodine is uh, attached to this tyrosine to form monoiodotyrosine. Two iodines are attached to this tyrosine to form diiodotyrosine. Then this monoiodotyrosine and diiodotyrosine, they can combine to form triiodothyronine. So now this becomes thyronine. So there are two phenyl rings attached. One molecule of monoiodotyrosine and one molecule of diiodotyrosine together forms the triiodothyronine. It is also known as T3. And if two molecules of diiodotyrosine combines, they form the thyroxine or tetraiodothyronine, and it is called T4. Okay, so the tyrosine molecules that are present in thyroglobulin, they are iodinated first to form the monoiodotyrosine and diiodotyrosine. Then, depending on if these two are combining or two molecules of diiodotyrosine are combining, we get T3 and T4. So thyroglobulin will have different T3 and T4 attached to it. And this protein thyroglobulin, it undergoes proteolytic breakdown to release this hormone T3 and T4. That these are the thyroid hormones, triiodothyronine and thyroxine, that is tetraiodothyronine. Is this clear?
Yes, ma'am. It's clear, ma'am.
Okay. Next question is iodine organification. Option A occurs in the luminal surface of the follicular cell. Option B requires the enzyme thyroreductase. Option C requires the enzyme deiodinase. And option D requires copper as oxidizing agent. Iodine organification. Option A occurs in the luminal surface of follicular cells. Option B requires the enzyme thyroreductase. Option C requires the enzyme deiodinase. And option D requires copper as oxidizing agent. You are saying option A, that is iodine organification occurs in luminal surface of follicular cells. That is the correct answer. So what is iodine organification? It's basically the addition of iodine to the tyrosine molecule, phenyl ring of the tyrosine uh, that are present in the thyroglobulin protein. So you saw the thyroid hormone synthesis in the previous slide, right? So for the iodine to get attached to the tyrosine residues, we need the iodine to be transported first. So from the extracellular spaces, the iodine are transported by iodine transporters. So these are sodium potassium ATPase dependent thyroidal iodine transporter, also known as TSH. So these transporters, they transport the iodine from the extracellular space inside the thyroid cell, and then these iodine are taken to the follicular space, luminal surface of the follicular space, where iodine organification occurs. And iodine is oxidized and attached, and then iodination starts, where monoiodotyrosine and diiodotyrosine form. So you can see MIT and DIT are written. So the tyrosine residues that are present on thyroglobulin, they get uh, iodinated, and then coupling starts. So two molecules of diiodotyrosine together can form T4, that is thyroxine. One molecule of MIT and one molecule of DIT can form T3, that is triiodothyronine. Okay. So you can see here tyrosine residues on the thyroglobulin, they can either form monoiodotyrosine or diiodotyrosine. Then these MIT and DIT can together form T3, and DIT, two molecules of DIT can form T4. So the thyroglobulin will have the T3 and T4. Once these are present on the thyroglobulin, they will be phagocytosed inside the thyroid cell. So this is a process of uptaking the outer molecules. So the membrane will pinch in this thyroglobulin protein, form a endosome. This endosome will fuse with lysosomes to form endolysosomes. And inside that lysosome, so lysosome has slightly acidic environment where and it has many proteolytic enzymes. So in the endolysosome, there can be proteolysis where the T3 and T4 will be released. These T3 and T4 will be released outside the thyroid cells. The remaining MIT and DIT will be released, which will be deiodinated. So that iodine can be recycled, the tyrosine will be released out, and again the whole process can keep going on. Okay. Is this clear? If you have any doubt, let me know.
It's clear, ma'am.
Okay. Then we can move to the next question. The basic pathogenic mechanism of albinism are all except option A, deficiency of enzyme tyrosinase. Option B, decrease in melanosomes of melanocytes. Option C, impairment in melanin polymerization. Option D, limitation of substrate tyrosine availability. And option E, overactivation of protein matrix in the melanosome. Basic pathogenic mechanism of albinism are all except deficiency of the enzyme tyrosinase, decrease in the melanosome of melanocytes, impairment of melanin polymerization, limitation of substrate availability, that is tyrosine availability. Option E is overactivation of protein matrix in the melanosomes. So what is the which of the following is not responsible for albinism basically? Deficiency of enzyme tyrosinase, decrease in melanosomes of melanocyte, impairment of melanin polymerization, limitation of the substrate availability, and overactivation of the protein matrix in the melanosomes. You are saying the option E, that is overactivation of protein matrix and melanosome. That is the correct answer. So rest all the other four options can lead to albinism. So tyrosinase is the enzyme that converts tyrosine into melanin. Melanin is responsible for the uh, development of this color. Melanin is the color pigment basically. So the skin color, the hair color, our eye color, everything comes from melanin. Okay. So, and it's a polymer. Uh, so tyrosine, like in the dopamine synthesis, in this melanin biosynthesis pathway also, tyrosine is converted into dopa first. But in this case, the enzyme is tyrosinase. It's a copper containing enzyme. And this tyrosinase enzyme is actually present inside the melanosomes of melanocytes. So melanocytes are the skin cells. And inside the melanocytes, we have melanosomes. These are endosomal structures. And inside this melanosomes, we have the tyrosinase. It's a membrane protein. So tyrosinase protein is attached to the membrane of the melanosomes. Okay. And this melanin is secreted in the melanosome and like synthesized in the melanosome and secreted out in the melanocyte to give the color to the skin or give the color to the hair. Okay. So inside the melanocyte, tyrosinase is present in the melanosome membrane, and tyrosinase converts the tyrosine into dopa. Then dopa is converted into dopaquinone and further cyclized to dopachrome. Dopachrome then gets converted into indolic compounds, dihydroxyindole or dihydroxyindole carboxylic acid. And these compounds can polymerize to form the eumelanin. So this is the eumelanin pathway. So melanin are also of two types. We have eumelanin and pheomelanin. Eumelanin is responsible for the black and brown color, and pheomelanin is responsible for the red and yellow color. Okay, so basically the different wide variety of skin color that you see is due to the presence of both eumelanin and pheomelanin at different ratios. So that's how we get the different races have different skin color, and different people have different hair color, natural hair color I'm talking about. So that is due to the presence of eumelanin and pheomelanin at different ratios. So in different individuals, the amount of melanin content is different, okay? And the structure of melanin also differs. So tyrosinase converts tyrosine into these melanin pigments, and lack of these pigments can lead to albinism. Albino means white. Okay. So if the pigments are not forming, then there won't be any skin color. So you might have seen uh, cases of vitiligo where certain patches of skin turns white. In case of total albinism, the whole skin, the complete body is turns white, turns pale. There's no melanin, and it's actually it can lead to certain serious problems, like it can lead to melanoma. So melanin acts as a UV protector in our skin, so it can absorb the UV radiations, and it can protect the skin from UV damage. And if melanin is not synthesized in the body properly, like in case of albinism, they are very much prone to skin cancer. Okay, melanoma. So the lack of melanin in eyes can lead to ocular albinism. Lack of melanin in the skin as well as an eye can lead to oculocutaneous albinism. So there are two types of conditions: ocular albinism and oculocutaneous albinism. So basically, deficiency of the enzyme tyrosinase can lead to albinism because there won't be any melanin formation. Decrease in melanosomes. So if there are less melanosomes, automatically the amount of tyrosinase will be less and melanin would be less. Impairment in melanin polymerization. So I told you these indolic compounds, they polymerize to form these melanin. And in case of pheomelanin, cysteine dopa polymerizes to form pheomelanin. So this polymerization, if there's impairment in the polymerization, then also it can lead to albinism because there won't be any melanin formation. Then limitation of the substrate availability. If tyrosine itself is less in the body, like in case of phenylketonuria, since tyrosine is less in body, in case of phenylketonuria also, pale skin or white skin is observed, lighter skin is observed because there's a lack of melanin synthesis. Okay. So basically limitation in tyrosine can lead to albinism as well. The other manifestation of melanin biosynthesis pathways in case of overproduction of melanin. What will happen in that case? Overproduction of melanin can actually lead to hyperpigmentation. So dark spots that are present on our skin, they are actually due to overproduction of melanin. Okay, so these are the cases where the melanin production is low. That is underproduction of melanin. In case of overproduction of melanin, we have cases like hyperpigmentation. So if you have seen these skin creams, they have compounds like arbutin or kojic acid. So all these kojic acid, alpha arbutin, they are actually inhibitors of tyrosinase enzyme. So they can inhibit this enzyme, leading to lower synthesis of melanin, and that can actually treat the pigmentation problem. So all these skin lightening creams have these pigments that can inhibit the tyrosinase. Okay. I hope that's clear. If you have any doubt in the melanin biosynthesis pathway, you can ask. Moving on to the next question. Biologically important substances produced from tryptophan are all except option A, acetoacetyl coenzyme A, option B, niacin, option C, serotonin, option D, hydroxyindole acetic acid, and option E, dopamine. Which of the following is not synthesized from tryptophan? Acetoacetyl coenzyme A, niacin, serotonin, hydroxyindole acid, and dopamine. You've answered option E, that is dopamine. That is the correct answer. So we have already seen dopamine is synthesized from tyrosine. Tyrosine is converted into dopa and then dopamine. All the other products, acetoacetyl coenzyme A, niacin, serotonin, and hydroxyindole acid, these are synthesized from or produced from tryptophan. We'll see how. So acetoacetyl coenzyme A comes under the catabolism of tryptophan. So tryptophan is broken down and the final product is acetoacetate. Uh, and acetoacetate can take up a coenzyme A to form acetoacetyl coenzyme A. So acetoacetate, as if you remember, it's a ketone body. It's a ketogenic molecule. So like tyrosine was broken down into fumarate and acetoacetate. In case of tryptophan, it is completely broken down into acetoacetate. So first tryptophan gets converted into N-formyl kynurenine, then by the enzyme pyrrolase. Then this gets converted into kynurenine by the enzyme formylase. Then by another hydroxylase, this kynurenine is hydroxylated. Then you have kynurenase, which hydrolyzes the molecule into 3-hydroxyanthranilate, and then ring opening occurs to form 2-acroyl 3-amino fumarate by the enzyme oxidase. Then decarboxylation occurs by an enzyme decarboxylase to form 2-amino cis-muconate semialdehyde, and this reduces to form ketoadipate, and then this can get converted into ketogluteryl coenzyme A, then acetoacetyl coenzyme A, and then acetoacetate. Okay. So acetoacetyl coenzyme A is one of the catabolic product of tryptophan. Other than that, tryptophan get converted into indole. How? So you know that tryptophan carries this indole group, right? So this indole group can be released from the tryptophan. So tryptophan can get converted into indole pyruvic acid by the same transferase reaction. So tryptophan can give its amino group to alpha ketoglutaric acid. So alpha ketoglutaric acid will become glutamic acid and tryptophan will become indole pyruvic acid. So this part is pyruvic acid and then you have a indole group attached to this. So indole pyruvic acid. Then there is a oxidative decarboxylation with the coenzyme TLP. So the carbon dioxide will be removed and you will be left with indole acetic acid. So in the acetic acid, one side chain of indole is there. Then again, another decarboxylation reaction, one more CO2 will be removed to form ketol. And this ketol, and then the third carbon will be removed. The CH3 group will be removed in the form of CO2 to form indole. This indole can get oxidized to form indoxyl. Indoxyl can get sulfate, can take up the sulfate group to form indoxyl sulfate, and finally indican. Okay, so this is another metabolic pathway of tryptophan. Synthesis of indole from tryptophan or synthesis of indican from tryptophan. Then comes the synthesis of niacin molecules. So, you know, NAD plus, NADP plus, so you have seen these NADPH, NADP plus in various reaction from the first session onwards, right? From carbohydrate metabolism to fatty acid metabolism to uh, protein metabolism. Everywhere you have seen different enzymes that are either NADP dependent or NAD dependent or NADH dependent. So from where are these NAD, NADP coming from? So these are coming from tryptophan. So tryptophan is very important molecule for the synthesis of these niacin dependent. So this has the niacin group, nicotinamide or niacinamide molecule. So that is the vitamin part of the any vitamin part of these molecules. Nicotinamide or niacinamide is the vitamin. And tryptophan is actually converted into these niacin molecules. So it is important in the synthesis of NAD or NADP plus. Then tryptophan is also important in the synthesis of serotonin. How tryptophan is converted into 5-hydroxy tryptophan by hydroxylation of the indole side chain. Then decarboxylation occurs of for this 5-hydroxytryptophan to form 5-hydroxytryptamine, or called that is also known as serotonin. And this is a very important hormone in the body. Okay, so tryptophan is responsible for the synthesis of niacin, for the synthesis of serotonin, for the synthesis of indolates, and for the synthesis of acetoacetyl coenzyme A or acetoacetate. Okay, is that clear? Moving on to the next question then. Which of the following is not an inborn error of phenylalanine or tyrosine metabolism? Which of the following is not an inborn error of phenylalanine or tyrosine metabolism? Option A, alkaptonuria. Option B, tyrosinemia. Option C, Hartnup disease. And option D, phenylketonuria. Which of the following is not an inborn error of phenylalanine or tyrosine metabolism? Alkaptonuria, tyrosinemia, Hartnup disease, or phenylketonuria? Which of the following is not an inborn error of phenylalanine or tyrosine metabolism? Alkaptonuria, tyrosinemia, Hartnup disease, or phenylketonuria? You have answered option C, Hartnup disease. That is the correct answer. So we have already studied alkaptonuria, tyrosinemia, phenylketonuria. These are all defects in different enzymes of the tyrosine metabolism or phenylalanine metabolism. So Hartnup disease is actually caused by a defect in the tryptophan metabolic pathway. So tryptophan is a neutral amino acid that is precursor of niacin. I told you uh, niacin, the NADP plus and NAD plus synthesis from tryptophan. Wasting of tryptophan results in niacin deficiency, manifesting as pellagra-like skin eruptions and neurological symptoms. So these are the manifestations of Hartnup disease. It can cause dermatitis, ataxia. It can also cause amino acid aciduria. And excess of tryptophan present in the gut is converted into indolic compounds, as we have seen, by the action of colonic bacteria, and these indolic compounds are conjugated in the liver and are excreted in the urine as indican. How is it diagnosed? It is diagnosed by urine analysis showing neutral amino acid aciduria except proline. So all the other neutral amino acids except proline uh, are secreted in the urine, excreted in the urine. And indican is also present in the urine because the tryptophan, excess tryptophan that is not getting metabolized into niacin, it is converted into indolic compounds, and these indolic compounds are converted into indican that is excreted in the urine. And for treatment, we have to take a diet supplemented by niacin because that vitamin will be deficient in this case because that vitamin is not synthesized by tryptophan. Okay. Any doubt? Moving on to the next question. Defective serotonin metabolism can cause all the following except option A, diarrhea, option B, hot flushes, option C, jaundice, option D, wheezing, option E, pain in abdomen. Defective serotonin metabolism can cause all of the following except. So, which of the following is not caused during serotonin metabolism defect? Diarrhea, hot flushes, jaundice, wheezing, and pain in the abdomen. Defective serotonin metabolism can cause all of the following except diarrhea, hot flushes, jaundice, wheezing, and pain in abdomen. You have answered option C, that is jaundice. That is the correct answer. So all the other symptoms are observed during serotonin metabolism defect. So it can lead to diarrhea, hot flushes, wheezing, and pain in the abdomen. Okay. So this was the last question in the session. So if you have any doubt in any of these questions discussed today or in any of the topic in this week's session, let me know. Do you have any question? Okay. If there are no doubts, then I can close this session. I'll probably take two sessions in the next week on Monday and Tuesday, and I won't be taking any session in the week after that. Okay. So you will get the email for the sessions accordingly. I'll uh, make sure that the emails are forwarded. Okay. So thank you so much for joining. See you next week. We'll take two classes next week. Okay. Thank you.